Alrighty. So this is our last topic before the exam. Little bit of chapter 9, interactive effects. So we've been learning about how organisms can deal with toxic chemicals that they encounter in the environment, and we've also been learning about various ways that we can measure the potential for hazard and toxicity and the interactions of chemicals and what happens when you have mixtures of chemicals and all that. So this is very much a continuation of those kinds of thoughts. So this chapter is really focused on understanding the concept of synergism. And so the chapter begins with an introduction to the concept of the synergistic ratio. And we'll get to what the synergistic ratio is and what synergism means in a minute. But generally speaking, when you encounter a mixture of toxic substances out in the world, because we very rarely encounter just pure toxic chemical singly all on its own. It's usually in some other substance, it's dissolved into something, it's interacting with other chemicals that might be near it, or it will be a pure mixture. And so as an example that I've added to the slide, this image of this weed killer bottle here. And so it says very helpfully on the front that 41% of this stuff here is glyphosate. And so the question is, what is the remaining 59%? The remaining 59% is that mixture in which you encounter the glyphosate. Some of those compounds are going to not have anything really to do at all. They're indicated very unhelpfully on this label as inert compounds, which I know is hard to see, really tiny, but it says inert compounds right about there. There's some other substances like salt. Sometimes these extra compounds are in there to sort of help the delivery of the target compound to make sure that in this case, for example, we're trying to get this weed killer, this glyphosate, we're trying to get this to be absorbed by plants so that we can kill the weeds. And so a salt has been added to sort of help induce that osmotic difference that's going to encourage plants to take up additional substances that hopefully the glyphosate will be taken up along with it. So generally speaking, we do encounter chemicals in a mixture type form, rarely a pure form. And what we are counseled to assume is to assume that these toxic mixtures will have an additive effect, that when there are more than one toxic compounds together in a mixture, that the baseline effect that we should assume from that is that each of those toxic compounds will provide an additive effect. That if we have 1 unit of compound A and 1 unit of compound B, we can expect 2 units of toxic effect, purely additive. However, oftentimes, the other compounds that we encounter along with some target compound of interest are not interacting in a purely additive way. They're often acting in a synergistic way. And synergistic means that the toxic effect of some additional compound enhances the toxic effect of the initial compound. So, for example, if we have our glyphosate, so glyphosate is a compound that interrupts amino acid formation, and if you can't form proper amino acids, then you can't make proteins. If you can't make proteins, then you can't do things like photosynthesis. So glyphosate interferes with protein production. And it's bad enough all on its own. If you were just, able to take like a needle and a syringe and inject the plant with glyphosate, you would affect its ability. But since we don't just inject plants with glyphosate, we spray it, we need some additional chemical like the salt that they've got indicated here. We need some salt to help the plant be tricked into taking up that compound. That salt then helps amplify the toxic effect. So if we just sprayed the compound on the plant, just pure glyphosate, we would get some low level of toxic response to the glyphosate. But by adding the salt, we can amp up the toxic effect of the same amount of glyphosate just by adding a little bit of salt or whatever other compound it might be. So our synergistic concept here says that we add some additional compounds, some new compound to increase the toxicity of our main compound focus. We can, of course, mathematically express this by looking at the, and we can I'm sorry, not only can we mathematically express this, but we can help to define it by looking at it in a mathematical way, where we can examine the median lethal dose, the LD50 of some compound alone, and compare it to the LD50 of that same chemical plus whatever compound it is as a synergist. So in this example here, we could look at the LD50 for glyphosate, and then we could compare it to the LD50 for glyphosate plus that salt. And that comparison of these two things is what we're going to call our synergistic ratio. So with the synergistic ratio, we can compare the concentration or the LD50 of the chemical alone, compare it to the LD50 of the chemical plus its synergist, plus its amplifier. You can think of the synergist as an amplifier. And that will tell us the nature of just how synergistic this additional compound is going to be. All right, everybody with me so far? Okay, so if some additional compound is going to be a synergist, i.e. a compound that amplifies the toxicity, then we would expect a synergistic ratio to be greater than 1. I think I have that backwards. I think that's supposed to be less than 1. I apologize. Fix that. I literally just reread this chapter of the textbook to make sure that I had this right, and I think I missed that. Because now greater than 1 doesn't make sense. Oh, where am I going with this? My head is all scrambled today. I'm sorry. Well, let's throw up the rest of the slide and see if we can't unscramble my brain confusion. All right, so we're going to compare this to 1. So our median lethal dose of the chemical alone could be whatever it's going to be. It's going to be, say, 1 unit of toxic compound A, 1 unit of compound A. All right, there's our median lethal dose of whatever our particular compound is that I'm writing on the board. If the synergist is really going to be synergistic, i.e. amplify the toxic effect of that compound, then our denominator should be lower, i.e. we should need less of our original toxic compound, right? If the synergist amplifies the toxicity of our compound, then we need less of that actual toxic compound to get the same effect as the compound all by itself. Does that make sense? All right, and so then I think my original text is correct. Should be greater. Sorry. This is often how teaching yourself goes. You go back and forth with things looking at, gosh, that doesn't make sense. And you rework it, logic your way through it until it does make sense. Okay, so we have our toxic unit all by itself, unit of 1. And then if we need, say, 0.75 of compound A plus 1 unit of our synergist, well, 1 divided by 0.75 should be some number greater than 1. Yes? Because the median lethal dose of compound A with however much synergist, so we don't count the unit of the synergist towards this, if that makes sense, right? So we're not saying that our denominator here is 0.75 units of compound A plus 1 unit of synergist, i.e., and therefore 1.75 total in the denominator. We're really just looking at how much compound A do we need without a synergist in the numerator, with the synergist in the denominator. Does that make sense? Have I unscrambled everyone's head enough? Some nods, lots of blank stares. Okay. So, I'm sorry, say your question one more time. Yeah. Correct. No, because we're just focused on how much of the actual chemical you need. Right? So if you have, imagine you have that's not a flask. That's a flask. All right, so imagine we have a flask with just water in it. Right? And we add to it 1 unit of compound A. All right, so here's our pure chemical all by itself. All right, so now we're going to compare that and whatever, so this 1 unit, this equals our LD50. All right, so then we do some experiments. And so, all right, so just as a reminder, LD50, this is where 50% of our trial organisms die. All right, so pure chemical in water, 1 unit of compound A kills 50% of our organisms. Okay, so then now we have salt water. We're going to pretend that salt is our synergist. And we do a whole bunch of trials, and we find out that the LD50 of compound A, which is, remember, where 50% of our organisms are dying, we only need 0.6 units. So to get the same effect of LD50, we need 40% less with this saltwater synergist. And I left off the concentration of salt water to avoid confusion this time. Because as far as the synergistic ratio is concerned, the concentration of synergist that you need doesn't matter. Yes, we're just assuming that this. Correct. We're looking for what the LD50 is all by itself versus the LD50 with some synergists, like salt water, like sugar water, like diluted in oil or mixed into an oil mixture or mixed with any other compound. So we're not looking for the concentration of the synergist. We're looking for just how much of compound A, the toxic compound, do you need to kill 50% of your organisms with a synergist versus without a synergist. Does that clarify it for everyone else now? Okay. I'm seeing more head nods. Not all the heads nodding, but hopefully more people are along for the ride. And if not, then we can try again in a little bit. Okay, so we're looking for a synergistic ratio of greater than 1 to suggest to us that some toxic compound would benefit, more or less, quote unquote, benefit from having some additional chemical with it to increase its toxicity. And so as an example of some of that, here are some examples from your textbook. So we have some examples of pesticides like cypermethrin, carbonyl, and malathion. And then we add in some synergist, which for whatever reason, they've decided to also label it as inhibitor, which I agree is extremely confusing because if the synergist amplifies it, inhibitor sounds very counterintuitive. And I'm sure if we read the paper that they based this table on, that inhibitor statement would make a little bit more sense. But for our purposes here, I think it is safe to just ignore the word inhibitor because we're not looking to inhibit. We're looking for the effect of the synergist, the synergistic effect. So what this table is showing you is that with the additional synergist of piperonal butoxide, you can increase the toxicity of cypermethrin by almost 40 times. The little less than is also confusing. It's not saying less than 40 times toxic. It's saying that it's increasing the toxicity just shy of 40 times more toxic, meaning you need 40 times less of your cypermethrin if you mix it with piperonal butoxide. So you go from needing 40 grams just straight up to only needing 1 gram. And especially if you're going to be spraying something out into the environment, you want to spray as little of it as possible. So adding a synergist can not only make financial sense because you need less of this cypermethrin stuff, but when this stuff escapes out into the environment, a lower concentration of this stuff is getting out into the environment, and it can be sort of worked through, metabolized, and broken down by UV radiation or whatever else is going to happen to it much faster, right? Because cypermethrin sounds like permethrin, which is a pyrethroid type compound, which comes from the chrysanthemum flowers, which gets broken down very easily in the environment through UV degradation because it's a natural compound. Does that sound familiar? Cool. All right, so we can use 1 gram of it as opposed to 40 grams of it. And when that 1 gram escapes out into the environment, we just let it sit in the sunshine, and sunshine will take care of 1 gram of cypermethrin just like that. Does that make sense? Okay. So I apologize for the initial confusion, but we worked it out. We logicked it through together. Okay. All right, so we're hoping to find some compounds with some synergistic effects, but additive is a thing that we do still need to consider. So when compounds do exhibit some additive effect, usually this is a simple cumulative effect. So if you have various chemicals that whatever concentration dose of it will give you 5% mortality, another compound that you add to the mixture. So to come back to my drawing over here that hopefully everyone can see, but I don't think will be captured on the video. Ugh. So let's say we're just making a witch's brew. We're just making a witch's brew of substance. So we add into this compound A with some concentration that gives us a lethal dose of 5%. And then we similarly add to this some concentration of compound B that gives us an LD of 10%. And we add to this compound C at some concentration that gives us a lethal dose of 15%. So now this total mixture, this witch's brew, would have a total lethal effect under simple additive cumulative thought processes. This total potion here would give us an LD of 30% under additive thought processes. So some compounds do exhibit additive properties, which is different than synergistic, but a little bit easier to think of. We typically observe additive effects when all of the compounds that we're adding into our witch's brew, when all of those compounds have some similar mechanism or action or work on the same pathway. So for example, if compound A, B, and C in my little diagram over here, if all 3 of those compounds were acetylcholinesterase inhibitors, they're all acting on the same thing. They're all acting on that acetylcholine compound that will break apart acetylcholine as our neurotransmitter, acetylcholinesterase. So if everything here works on acetylcholinesterase, then they all have the same target, and so we would all expect them to kind of work in the same way at about the same rate, and so we can observe an additive effect here. Of course, we rarely see purely additive effects because there's always going to be differences. There will always be differences due to toxicokinetic factors, right? Compound A might be slightly more polar than compound B. So compound A will get metabolized and broken down faster. Compound B might be highly lipophilic, and so it's going to take a little bit longer to break down. So depending on the exact nature of all the compounds, we rarely see an actually purely additive effect. But we can generalize and say additive is something that exists. Does that make sense? All right, so one example of places where that works is in our vitamin K clotting cycle. So there are a number of compounds that can interfere with our vitamin K and blood clotting cycle. We've got warfarin, which we saw as a rodenticide, heparin, which very common drug, prescribed all the time, used in all sorts of medical applications. Phenadione, used in similar circumstances, and this brodifacuum, which is another one of those rodenticide anticoagulants that I showed you way back when we talked about chemical class compounds. So I'll just throw all these up here so that we can write and talk at the same time. So warfarin acts by preventing the activation of vitamin K. So in order for your blood to actually clot, vitamin K needs to be turned from its inactive into its active form so that the active form can go off and set off your cell signaling cascade of clotting factors. And so if you can't activate vitamin K, then it never turns on the cell signaling cycle of clotting factors. Warfarin inhibits the activation of vitamin K. Similarly, as does phenadione, and similarly, as does brodifacuum. So if you were exposed to equal parts warfarin and this phenadione, however you say that, if you were exposed to equal parts warfarin and phenadione, then you would experience exactly twice the amount of inactivation of vitamin K in an additive model. But we can see that warfarin versus phenadione, these are slightly different compounds. Warfarin's a bit bigger, has a few more non-polar, it's bigger, so it's going to take more time, but it does have a few more polar aspects. So it might get broken apart more easily because it has more sticky oxygens for your monooxygenase enzymes to latch onto. Phenadione has fewer oxygens, fewer sticky bits, and so it might take a little extra work. It might take a few rounds through phase 1 metabolism in order to break down the phenadione. And then we compare that to brodifucam. So if you had 1 part brodifucan and 1 part warfarin, again, you might expect 2 units of anti-activation of vitamin K with equal parts brodifucan and warfarin. But brodifucam, however you say that, because it's so much larger a molecule, has many fewer sticky bits, it's going to take even longer for your metabolism to try and break down. And so we might see slightly more than an additive effect because this is such a tricky compound for our bodies to break down. But we can see these examples of additive effect when we look at compounds that all have the same mechanism of action. They're all trying to inactivate vitamin K or similarly work on a different part of the same system. So heparin is going to interfere in here. It's going to inhibit the production of antithrombin. So if you were exposed to warfarin and heparin, you might still experience some additive effects because you have 2 different compounds interacting. Turn my laser back on. You have 2 different compounds interacting on 2 different parts of the same overall cell cycle process. And so they're attacking the whole cycle in 2 places instead of just one twice as much. Does that also sound okay? Okay. All right, so if only the world were as simple as additive, but much more likely we are going to have these synergistic effects, especially because the world exists in complex mixtures that we sometimes don't know exactly the components of. And so when we end up with a mixture, mixtures are typically more than additive and often become synergistic. So here in this diagram, we're looking at two different basic ways of thinking about how compounds can interact with each other. So on our x-axis, we have the composition of the mixture. So at this far end, our hypothetical mixture of compound that we are exposing some organism to is going to be all compound B, no compound A. At this other end, it's going to be all compound A, no compound B. So in the middle, right? So this is sort of like 50-50. 50-50 compound A, compound B. So you can make these different mixtures. You can make up different concentrations of compound A, right? So 0, 25%, 50%, 75%, different compound concentrations of compound B, 25%, 50%, 75%. Mix them both together so that you have 100% of all the compounds. And if you were to then give all of these different mixtures of 25-75, 50-50, 25-75, 100-100, you give all these mixtures to some group of organisms and you get the same LD50 every time, then that suggests that the effects of those are additive, because you always end up with the same total concentration of 100% toxic compound. You're just varying the amounts of A and B, and you get the same lethal effect every time. Does that make sense? All right. So if these compounds were additive, if they were additive, you get the same effect no matter what concentration we're looking at. Now, if you make your different concentrations of A and B, 0, 25, 50, 75, 100, 0, 25, 50, 75, 100. If you make those different concentrations and you get a greater or less than effect, lethal effect, and you can see this most clearly at the 50-50 mark. So at the 50-50 mark, so let's say you add them together, you've got equal concentrations, 50-50, and you get the highest amount of lethality you've ever seen at a 50-50 mark. This suggests that the interaction between compound A and B results in some synergism, and we can say that compound A or compound B is potentiating. So the potentiation is the enaction of that more than additive effect. So potentiation is where we see a more than additive effect. Similarly, if we see less than, we can call that antagonism. So if the combination, the equal combination 50-50 of our two toxic compounds results in a less than, then we know that these two compounds are fighting each other. We don't necessarily know which one is fighting which one, but we could see some antagonism where one compound is inhibiting the other. But when you have a synergistic interaction, you will see that two different compounds will potentiate each other. So the potentiation is the verb that makes synergism happen. There are two different mechanisms by which potentiation can effectively happen. So one mechanism is where compound A is going to inhibit some enzyme that detoxifies compound B. So remember, we saw additive when everything was working on the same pathways. With synergism and the potentiation that comes with synergism, we have two different compounds working on different parts of the physiology. And so by working on different parts of the physiology, that's now two battle fronts that your body has to fight and deal with. You have to deal with the toxic compound A and all of its peculiarities and predilections that come along with it for the way that its toxicokinetics are working, compound B for all the same reasons, and you might be having to do it on different battlefronts. Not everything's going to be attacking the same cell cycle, the same cells, the same part of your body, right? You could have ingested one, you could have breathed in another, you could be exposed to them in different ways. So with potentiation, you've got this battle on two fronts. So compound A could inhibit some enzyme that detoxifies compound B. If compound A is turning off your phase 1 metabolism, that's good news for compound B. Another way that we can see potentiation is if compound A induces some enzyme that activates compound B. So if you just had compound B and it was kind of meh, and then compound A really ramped up your monooxygenases such that when they break apart compound B, it turns it into an even more toxic form. You can similarly see some additive potentiation. If we're turning compound B from something non-toxic into something toxic, that is an example of potentiation, especially if compound A is the one doing it. And so we can make these mixtures, in a very simple way with just two compounds and vary their overall concentrations. Or we can look at more heterogeneous and complex mixtures that exist out in the natural world. All right, so does potentiation make sense? Okay. All right, so let's look at how each of these mechanisms could possibly come to be. So first we're going to look at potentiation due to inhibition. All right, so remember, we're looking at how these compounds are interacting in a body. So we have compounds being ingested, and then they can do a number of things within the body, and then ultimately they are excreted. And our metabolism is the way in which we try to break down these toxic compounds. You might recall that our metabolism exists in two phases. Phase 1, which uses those monooxygenase enzymes to break apart compounds, especially if they're lipophilic, add sticky hydroxyl groups to it. And then phase 2, which is more further digested by many and numerous and varied enzymes. All right, so remember, lipophilic compounds need to be broken down into more water-soluble form. Usually we're doing this by adding hydroxyl groups. This breakdown of highly lipophilic things is happening in phase 1 due to our monooxygenase enzymes. And so if you prevent phase 1, right, if you stop this, if you prevent phase 1, then it makes it really, really hard for phase 2 to happen. This doesn't necessarily turn off the enzymes in phase 2 that, if you don't have properly functioning monooxygenase enzymes, and you don't get the enzymes later on in phase 2. That's not what this is saying. What this is saying is that if you turn off the enzymes in phase 1, it makes the job of the enzymes in phase 2 harder, if not impossible. Right? If the enzymes in phase 2 are looking for hydroxyl groups and no hydroxyl groups were added, then phase 2 has a really hard time moving forward. And so if phase 2 can't move forward, then that means that toxic compound is just running around wherever it's running around, and you can't then waste it out. All right, so let's look a little bit more at these monooxygenase enzymes. All right, so again, the main function of these enzymes, monooxygenase enzymes, the main function of these guys is to add hydroxyl groups. This is a hydroxyl group. This adds polarity, it adds charge, it adds a sticky place for other enzymes to latch onto. It also helps to make the original compound smaller because usually you're breaking it to add that hydroxyl. Smaller compounds are easier to digest. Our monooxygenase enzymes generally serve two main functions. So other than the metabolic breakdown of toxic compounds, we see monooxygenase enzymes operating a lot in various signaling pathways. Because they are basically braking enzymes, they serve to reduce or inactivate other signaling molecules, turning things off, right? Acetylcholine isn't our only signaling molecule. We have all sorts of signaling molecules out there. And slapping hydroxyl groups on them makes them sticky. They don't want to interact with lipid membranes, makes it hard to transmit that signal, stops the signal entirely. One place that we see this is in the arachidonic cell signaling pathways. So the arachidonic cell signaling pathways are, they're an inflammatory stress response pathway. They're called arachidonic because they are primarily, they're the base signaling molecule that turns these pathways on comes from arachidonic acid, which we just sort of abbreviate as ARA. It's a fatty acid compound that has, let's see if I can remember the formula. It's got 20 carbons. It's got 4 double bonds throughout it. And the last double bond occurs at the 6th carbon. So it is an omega-6 fatty acid. So arachidonic cell signaling pathways are turned on by this arachidonic acid. Most fatty acids on the very last carbon, they end in a carboxyl group. So here's our last carbon. We can go this way and have an OH. We can go this way and have a double bonded oxygen. So this carboxyl group, this is the end, the tail end of most fatty acids. But if you are able to remove that oxygen, so it's just a carboxyl group, or if you have a methylated fatty acid, so that it's a methyl group at the end that is extremely lipophilic, and you remove that methyl group so that you have just a hydroxyl group, then you are able to interrupt how various proteins and plasma membranes are able to receive arachidonic acid and interact with that cell signaling pathway. And so it's monooxygenase enzymes that start to break down arachidonic acid and turn off your inflammation stress signaling pathways. All right, so that's one way in which monooxygenases work. And then the way that we've been focused on here is in toxic metabolism, where they're doing the same thing, but kind of inside a cell rather than maybe outside a cell. So monooxygenases are involved in phase 1 metabolism. And by throwing all sorts of hydroxyl groups on them, we can prevent toxic compounds from getting intermingled and tangled up in our cell signaling pathways. And so here's one particular toxic compound, and we can see that there are monooxygenases involved in a couple of steps in the breakdown of this whole compound. Right, so this is the whole compound, and we have monooxygenases first interacting with this part to break apart these rings from this guy right here. And now we have everything broken apart. And so we can start to throw all sorts of sticky bits. And at the end of the day, we end up with this rather benign compound and this slightly less toxic working its way towards benign compound. And so if we can potentiate by inhibiting monooxygenases, then our compound here, permethrin, our compound here stays whole, and permethrin keeps circulating throughout your body if you turn off those monooxygenase enzymes through some potentiation. All right, so permethrin's a tricky one because you need 2 different rounds of monooxygenase to get it broken down enough to go on to phase 2. And so if you can potentiate permethrin by turning off the monooxygenases with some other compound, then the permethrin sticks around longer and does more of its damage. Does that make sense? All right. All right, so that's potentiation due to inactivation. And so now we can see potentiation due to inhibition. That was the word that we used, inhibition, inhibiting of monooxygenases. And so now we can look at potentiation due to activation. So here, the monooxygenases get to do their job, but the thing that the product of that monooxygenase metabolism is itself toxic. So one example of that is benzopyrene. So benzopyrene all by itself is not toxic, but monooxygenase enzymes, when they try to break this down, you end up making dioxin. And so benzopyrene isn't of itself toxic. The reason why benzopyrene is toxic and you shouldn't consume it is because once it gets into your body, you will make it into dioxin. So I'm not saying go out and eat benzopyrene, you're fine, because you can't tell your cells, don't break apart the benzopyrene, just let it go. Because your cells are just going to do that without you having any control over it, and they're going to make dioxin out of benzopyrene. Yeah, so these are, monooxygenase is phase 1. That's all that was there. All right. Within the monooxygenase enzyme group, there is a particular collection of monooxygenase enzymes that we have been introduced to before, but we're going to talk a little bit more now. And that is the cytochrome P450 family of monooxygenase enzymes. So there's all different monooxygenase enzymes. And when your textbook talks about monooxygenases, it's talking about all however many dozens of different kinds of monooxygenases there are. But in particular, one group of monooxygenases are the cytochrome P450 enzymes. So these enzymes are monooxygenases. They do a lot of this stuff, like turning benzopyrene into dioxin. And one place where they particularly shine is in organophosphorus insecticides. They're really good at finding these phosphorus double bonded to sulfur groups. So here, for example, so here in this organophosphorus compound, they're going to look for that, and they're going to turn it into a phosphorus double bonded to an oxygen. An oxygen is much more sticky and able to be enacted upon by phase 2 compounds than sulfur. But generally speaking, these cytochrome compounds are really good at oxidizing. Just like we see monooxygenase enzymes involved in other areas of our cellular metabolism, cytochromes are also involved in other areas of our metabolism. They are present in familiar cellular processes. And so I've got chloroplast here with the whole, right here's our whole Z scheme. Remember the Z scheme from photosynthesis? I'm not going to ask you to know the Z scheme or know anything about photosynthesis. It's just here for you to see. So here's photosynthesis, Z scheme. Here's the electron transport chain in our mitochondria, make an ATP. And we see cytochromes in both of these. So cytochromes are really important molecules, enzymes, compounds, whatever you want to call them. So there's a whole bunch of different cytochromes. They're not all the P450 variety of cytochromes, but cytochromes generally are compounds that exist. Cytochromes are used all over our bodies. And just like our cytochrome P450s in its monooxygenase capacity, these cytochromes here are doing something very similar. They're oxidizing. So cytochromes here in photosynthesis and electron transport chain, right? We're moving electrons along, reducing electrons all the way on down. Same on both of these, so we're oxidizing. And P450s are also oxidizers. We're good at oxidizing, particularly those xenobiotics, those really weird compounds. They're also really important in the synthesis and metabolism of hormones and various hormone mimics. So plastic-like compounds, synthetic hormones, phthalates, all those various hormone mimics. So while cytochromes are all over the place in many and varied forms, particularly of the P450 variety of enzymes, we have 57 unique cytochrome P450 enzymes that can be involved in the oxidation of our toxic compounds. So cytochrome P450s are really the workhorses of the monooxygenase group of compounds. All right, everybody good here? Okay. But there are lots of compounds out there that can inhibit and impair cytochrome P450s. So CY is our abbreviation for cytochrome, so that you don't have to write cytochrome every damn time. We just call them CY P450s. All right, so we can get alterations. Cytochrome P450s can alter. They themselves can be altered, and they can make weird alterations to compounds that they're trying to break down. So some drugs can inhibit cytochrome P450s, and other compounds can promote cytochrome P450s to break things down in a way that is not their original function. Especially this promote the breakdown of drugs to alter their effect. We can see this inadvertently creating new, that's what novel means. Novel means new toxic compounds. And so here's a whole paper here. If you wanted to read this paper, this is about how your cytochrome P450s can take phenammophos and break it down. There are all the ways they can break it down correctly and all the ways that they can break it down and make accidental toxic compounds. So even for one compound, if you interact your cytochrome P450s with other potentiating compounds, things can go haywire. And potentiating compounds don't have to just be other toxic things. There are plenty of benevolent inhibitors out there that especially act on cytochrome P450. So grapefruit juice is kind of the classic one. There's a compound in grapefruit juice that inhibits your body's ability to break down certain heart medications into their functional form. There's like a whole suite of heart medications that the actual form you ingest is not the active form. You have to wait for your body's metabolism to break it down into the actual active form of those heart medications. And if you take that pill with a glass of grapefruit juice, you inhibit your body's ability to turn that inactive drug into the active form of the drug. So like my mom is on a heart medication, she could never drink grapefruit juice again. The whole like other citrus that you could make margaritas with is out the window for her, especially palomas, which have grapefruit juice in them. It's totally a non-starter. Bergamot is another one. So bergamot is another kind of citrus, right? So grapefruit is a citrus, bergamot is a citrus. Bergamot is the citrusy flavor that's in Earl Grey tea. So if you like your tea Earl Grey hot, then the thing that gives Earl Grey tea its particular flavor, it's not just dried black tea, it's dried black tea with bergamot. So no drinking Earl Grey tea if you're on certain medications. St. John's wort deactivates some of the hormones in over-the-counter birth control pills. So if you are on over-the-counter birth control pills, stop taking St. John's wort, because it can inactivate the birth control function of your birth control. I'm not sure how much that applies to if you have like a ring or an implant or you're on the Depo-Provera shot. But I know that for the tablet over-the-counter pill, or not even the over-the-counter pill, just the tablet form of most birth control pills, St. John's wort will inactivate your body's ability to use effectively that hormone compound. And then, of course, we have our usual toxic compounds that can inhibit or otherwise impair your cytochrome P450s, so tin tributyl, which we saw killing off all sorts of whelks in New England, carbaryl, organophosphates, etc. All right, so when are we likely to encounter potentiation and synergistic effect of compounds? Typically mostly in heavily polluted areas. So in heavily polluted areas, right, where you have all sorts of compounds existing in the environment, you're going to get them put together in all sorts of new and different ways. So new and different ways of things. Industrial areas, so if you were to be in an area of like Niagara Falls, where all of the chemical processing plants are, walking through, breathing the air. There was a story that I posted to the toxicology in the news. I don't know if anyone's opened that document on Blackboard, but there was a recent story about a Goodyear tire manufacturing plant in Niagara Falls that was releasing a particular compound into the air at levels above what the DEC and EPA said should be around. And so in those industrial areas, you've got this whatever toxic compound that Goodyear is releasing next to whatever other toxic compound that DuPont, to just pull a name out of the air, is releasing into the air, and you can get these two things interacting when they wouldn't normally be found together. Similarly with agricultural areas, where you've got insecticides and herbicides and other, whole collections of pesticides all going around, you get things combining and potentially potentiating. We usually can diagnose that potentiating is happening by testing individuals and finding elevated levels of monooxygenase enzymes is usually how we test that. And so one of the reasons why I mentioned that is that we might be seeing some of this popping up in the next several weeks following the aftermath of Hurricane Helene. So for those of you not following the news, we just had a major Category 4 hurricane land. It made landfall in the Big Bend area of Florida, but then continued inland for several hundred miles. The rain that we had Saturday night was some of the rain from Helene. So Helene, the effects of Helene made it all the way up here to us. But especially places in the Appalachian Mountains were really severely affected. And so just as a couple of examples, there's all sorts of devastating photos out there, but this is the town of Chimney Rock in North Carolina, in Western North Carolina in the Appalachian Mountains. Here's a very nice before picture of what it looked like, and now here is the after picture. So this building and all of its components, all of its insulation, all of its wiring, all of its coating, any of the chemicals stored in that building, any of the food that was in that building, any pesticides that might have been used in that building, all of these cars and all of their different fluids that make a car run, and the asphalt and all of the different compounds that go into asphalt manufacture and production have all been churned up into a soup and are now floating through this river as people try to, go through and recollect pieces of their lives. And even once these floodwaters subside, the remaining rubble will be coated in this soupy mixture from all of those things. As another example of what we can see, so this is the textile plant Celanese. Celanese is a textile manufacturing plant, meaning that they make fabric-y sort of products. And one of the things in particular that Celanese makes is they make the filter tips that go on filtered cigarettes. There's a whole plant for this. And I really only know about this plant because this is where my in-laws live. So this plant, so it goes all this way. And normally, this is the New River that goes through southwestern Virginia. Normally the New River's like over here. And so the river has flooded all the way. This is supposed to be like a nice park area with like a kayak launch and stuff. and like a parking lot and everything. And then there's the manufacturing plant. And you can see that the water is all the way into the manufacturing plant. It did not cross this rail track that goes here into the main area of the plant. It just affected these sort of outer bits of the plant. But, you know, cigarette filter tips are not the only thing they manufacture. It's just kind of the thing they're most famous for. So whatever chemicals are in, you know, here or here or here, we don't know. We don't know. There are other places where the river did swamp over the railroad tracks. And so if there was any coal being hauled or natural gas being hauled or oil being hauled or whatever it might have been on some of those tankers on a train car, that is now all in the river and being mixed and churned. And as people are trying to work through the debris as the river subsides, they will be exposed to all of these mixtures, these new combinations that aren't meant to exist, that we don't know what's going to happen when unknown concentrations of things in industrial plants are mixed all together. All right, and so with that, that's the end of our content. So as a very quick exam review, generally speaking, what's going to be on the exam? You should be familiar with the main categories of toxic compounds and be able to recognize their distinctive features, i.e. what is it that makes a pyrethroid, right? There's usually sort of two ring groups bridged together and then some crazy antenna bit, right? That's kind of what I'm looking for in identifying, for example, a pyrethroid. So I will show you chemical structures, and I will give you a chemical name word bank, and I will expect you to match. So hopefully that sounds doable. Rather than you just having to know the names, you can process of elimination your way through it. You should know key features like that pyrethroids are able to be degraded by UV, or that organophosphoruses are usually applied in a solid format as opposed to a liquid format like organochlorines, things like that. The factors that are going to contribute to the likelihood of a chemical being a problem. So it's lipophilic status, how KOW relates to its lipophilic status, how its lipophilic status relates to how it's going to be processed through the 5 stages of uptake, action, storage, metabolism, and excretion. Things like that. And then, and of course, other various concepts like bioaccumulation versus bioconcentration, synergistic ratio, the risk factor that we talked about last time, those sort of basic terms and concepts that we can use to describe things. Be aware of the different components of the environment, land, air, sea, how things are going to get into any one, how any one compartment might interact with another, right? So when we talked about metals being concentrated in water body sediments, and then those water bodies flood onto agricultural fields and deposit those metals, increasing the metal concentration of crops. Things like that. And then, you know, some examples as needed. All right, so that's all I got. So I will stop this recording, and if folks want to, if folks have questions that they want to ask, I'm happy to stick around and answer questions. If you have no questions, then I see no point in keeping you here. So we're going to stop the recording. There it is.